All right, we are going to finish this up with talking about the actual organelles in the cell. And then I'm going to assign you a Bozeman video and maybe a little activity. And then when we come back together in class, we're going to do a little group activity around this too, just see how you're doing with all of it. All right, so here's a picture of a plant cell, animal cell. I'd ask you if we were in the class together, and hopefully someone would say, that looks like an animal cell, and they would be right, because that is an animal cell. So in our animal cell, now that I have a cursor, this, see here, this part right here, let me get a pin. This part right here where I'm drawing a line, this thing right here, this is the cell membrane, right? And then I'm going to talk about these independently, but this is the nucleus right here. Inside the nucleus is like another nucleus. That thing is called the nucleolus. I'll circle it. Nucleolus. Sorry, I have a touchpad and it's hard to circle. Then we have, I was trying to see if I could use my iPad pin. Then this crazy looking thing here is called the Endoplasmic Reticulum. You'll see that word in a minute. This guy over here is the Golgi body. These guys are mitochondria. This is part of these strandy things are part of the cytoskeleton. These dots can be lysosomes. Talk more about them. There's like lysosomes and they're peroxisomes. They're kind of similar to one another. And then the little dots, all either by themselves. I'm going to circle a few of them. These guys, all these dots. Those guys are called ribosomes. You notice they're also stuck to the endoplasmic reticulum right there. Those are ribosomes. So we'll talk about the function of those things in a minute. So these are the organelles of the cell, right? They all have a job, and that's what we're going to learn about. The inside of the cell is filled with this stuff called cytoplasm, which is kind of watery, gelatinous watery, right? So it's not just like, it's like a gel-like watery substance. And all of those organelles are kind of suspended in it. It's like if any of you have ever had the unfortunate experience of having to eat one of those Jell-O salads, unless maybe you like them, but I find them kind of disgusting myself. But they're good. It's a good cell model. When my son was in school, we did a Jell-O cell, which was actually kind of fun. But when you put the grapes and the nuts and whatever other kinds of things people throw in those Jell-O salads, then they kind of suspend in the Jell-O. The Jell-O is the cytoplasm, and all the stuff suspended in there would be the organelles. All right, so let's start with the nucleus, arguably the most important part of the cell. Actually, I don't know that anyone really would argue that. So in the center of the cell is the nucleus. The nucleus is surrounded by a membrane. It's actually like a double membrane. So this membrane is called the nuclear membrane, or sometimes it's called the nuclear envelope. So that is the control center of the cell, right? So inside that nucleolus, that's a place where the ribosomes are made. So that is something that oftentimes they don't write about in books. And as I'm talking, I'm looking through your book. Let's see here if they talk about it. They label it, but oftentimes they don't. Oh, they do talk about it. Oh, good. So they mention it. So embedded in this nuclear envelope, inside here is the nucleolus. Okay, so again, the nuclear envelope is a double membrane, which you can see there, two-sided. And it has these, it's like kind of punctuated by these pores. And these pores are important because things can get through them. Ions, molecules, and specifically RNA, which we're going to learn about later. So when we learn about cell division and protein synthesis and all that, when RNA leaves the nucleus, it's going to come out of those holes. All right, the other thing, see all these sort of stringy things in here? That stuff is called chromatin. Chromatin is basically what our chromosomes are made up of. So DNA and proteins kind of all sort of like strung together is called chromatin. That chromatin will wind up really densely right before a cell divides, and then we'll call that stuff chromosomes. They're kind of linear structures at that point, right before the cell divides. But when the cell is not dividing, the chromatin is kind of spread out all over the place. So the chromatin, the important thing to note here is that all the genetic information, all of your DNA, is inside the nucleus, and we don't want it ever coming out. So that's part of the role of the nuclear envelope, is to keep the DNA inside of there, right? So the nucleus does direct the production of ribosomes, and basically that happens inside the nucleolus. So the nucleolus's job is to kind of aggregate the ribosomal RNA and, complex it with proteins and make these things called ribosomes, which will then leave the nucleus out of those little holes as well. Okay, so that was kind of a lot. So normally in a class I write on with my pen, but I can't do that on this. So the nucleus is the control center of the cell. I'm just giving you a quick summary. The control center of the cell, it's wrapped in a double membrane. Inside of the nucleus, all that stringy stuff that's colored white is chromatin. That houses your genetic information, your DNA, and some other proteins and stuff we'll talk about later. The innermost part is the nucleolus, where basically the RNA kind of gets like built up and made into ribosomes, or aggregated maybe together and made into ribosomes. And then those ribosomes get transported out of there. Okay, so I'm going to use my little pin to circle the nucleolus for you. Maybe blue. All right, so I'm circling the nucleolus. I'm not circling it very well because my finger is not very good at drawing. All right, so that's the nucleolus. I'm going to sort of trace some chromatin in here. So all this stuff that looks sort of like thread, that's all chromatin. This is the nuclear envelope right here. And then these are the pores. Okay, so and this we could call, if I were to ask you, what's the control center of the cell? You would tell me nucleus, hopefully. Where do ribosomes get made? You would tell me, don't say in the nucleus. I mean, that's true, but more appropriately in the nucleolus. That's what it'd be after there. All right, so here's a picture of this is basically a chromosome, and then looking at it really closely, see all these, like, it's just strands of information kind of wrapped around this blue thread. So these proteins are called histones, and so this is the DNA, right? So the DNA gets wrapped around these histone proteins, which are like spools, and this is like thread, and then they all kind of get stuck in here, and then they ultimately make this crazy structure, which is called a chromosome. So we'll talk more about that later on. All right, let's move outside of the nucleus to the closest organelle to the nucleus, which is called the endoplasmic reticulum. And it's basically made up of the series of interconnected membrane tubules. So tubes made out of membrane. The job of the endomembrane system, or the endoplasmic reticulum, is collectively to modify proteins and make lipids. All right, so these are the two main jobs of the endoplasmic reticulum: to modify proteins and synthesize lipids, or to make lipids. They're made in different areas of the endoplasmic reticulum. So this area right here, this area right here that I'm going to try to circle over here, look at it, looks different than this part. So what's different about this versus this? Well, hopefully you're saying, well, this part has bumps on it. Yeah, that's the difference. So what are these bumps called? Does anybody remember? Those bumps are called ribosomes, which are made in the nucleolus, pardon me. So the ribosomes that are made in the nucleolus, and they squeeze out of these pores, some of them get stuck to the endoplasmic reticulum. Some of them just float around in the cell. We've seen that before. Some do not. There's one part of the endoplasmic reticulum that does not have bumps, and there's one part that's bumpy. The part that's bumpy is called the smooth ER, and the part that's bumpy is called the rough ER, or endoplasmic reticulum. The smooth ER makes lipids. This is going to look like a monkey is drawing because I'm using my trackpad. Lipid. Smooth ER makes lipids. This part, the rough ER, I can't write that, but it synthesizes proteins. And you'll notice they're right on the, it's almost contiguous with the endoplasm, with the nucleus. So right on the other side of the nucleus is the endoplasmic reticulum. And it's actually like the phospholipid bilayer of the endoplasmic reticulum is actually continuous with the nuclear membrane. Like you can see it here. It's continuous with the outer phospholipid bilayer of the nuclear membrane. So they are actually connected. All right. The hollow part, you'll see the hollow part. That hollow part is called the, excuse me, the hollow part is called the lumen, or sometimes it's called the cisternal space. You don't need to remember that, but you'll see it in your book probably. All right, so the rough endoplasmic reticulum is called rough because when you look at it under a microscope, a really high power microscope, it looks bumpy. And the reason why it's bumpy is because it has ribosomes, and ribosomes, the job of ribosomes is to make protein. So let's flip it. So here's a ribosome, and this is the little thing that's happening at the top. It's making a protein. This is a protein, primary structure. Remember what primary structure is? Just like a strand. Whoops, sorry. So this, what you're seeing here, ribosomes are made up of a couple subunits, which we'll talk more about later. This strand, this is actually ribosomal, this is actually RNA, messenger RNA, which is going to come from the nucleus, and it's going to come out of the nucleus, and it's going to squeeze through these two subunits, and it's going to get read by this ribosome, and then this ribosome is essentially going to, with the help of another type of RNA, it's going to make a protein. So ribosomes make proteins. So when we go to our endoplasmic reticulum, right, and I say that the rough ER synthesizes proteins, it does that because of the ribosomes. That's why it's making proteins. If it wasn't for the ribosomes, it would not be able to make proteins. So really, it's the ribosomes that are making proteins. The rough endoplasmic reticulum is just the site of one of the sites of protein synthesis because it's the ribosome that's actually doing the job. Okay, so let's see, what else do I need to tell you about that? So ribosomes are going to be the ones that synthesize the proteins when they're attached to the rough endoplasmic reticulum. And then those proteins that are made are going to move into the lumen, the inside, that hollow space of the rough endoplasmic reticulum, where then they're going to get further modified. So the other side of the endoplasmic reticulum, the smooth part, that part's going to make phospholipids, mostly for the cell membranes. That's where that stuff comes from. So the phospholipids, or the proteins, they don't stay in the rough endoplasmic reticulum. They get packaged into these little kind of bubbles, which we call vesicles. And from there, they go someplace else. And one of the places that they can go is to the Golgi body, which I think is what I have next. Yes. So the Golgi body, or sometimes it's called the Golgi Apparatus. It's also located near the endoplasmic reticulum, and it essentially has a face. It faces the endoplasmic reticulum, the receiving face, and then it has a releasing face on the side away from the endoplasmic reticulum towards the cell membrane. And so the vesicles that were packaged in the endoplasmic reticulum travel to the receiving end of the Golgi body. They fuse with it. They dump their contents into the Golgi apparatus, and then the proteins or the lipids, depending, they kind of get passed through the Golgi body and they get modified even more. generally what's happening is they're adding like sugar chains to it. You know, they're adding groups, small molecular groups to those proteins or lipids. And then ultimately the Golgi body is responsible for routing that package that's, you know, some kind of modified protein or a lipid to someplace else, right? So they get kind of repackaged and then routed to their ultimate destination. So, you know, different cells are going to have more Golgi bodies than other cells. Cells that make a whole bunch of stuff, you know, like cells that make enzymes or, you know, antibodies and those kinds of things, have a lot of Golgi bodies. If they don't make a whole bunch of stuff, if the cell doesn't make a whole bunch of stuff, they won't have as many Golgi bodies. So here's a little picture of the whole, and we're going to learn this in a lot more detail later on, but these are all the things we've talked about. So this is the nucleus. This is DNA. DNA never leaves the nucleus. So, but it has all the information. It has all the important information. So the information from the DNA has to get out of the DNA, and it has to get put into a form that it can leave the nucleus. So it turns out that, and it's kind of crazy how it does it, but basically this is going to unzip and we're going to copy it to a strand of RNA. This one's called messenger RNA. Don't worry about all the details now. You'll learn it more later. Messenger RNA squeezes out through the pores and goes to find a ribosome, of which there's many all over the place. So let's just say there's a ribosome right here. It's going to wedge in the ribosome. The ribosome's going to build the protein based on the message, based on the code that came from here, got copied to this, then it gets translated out here. So they call this, when you copy the message from this to this, they call it transcription. And then when it gets basically made into a protein, it's called translation. So the message gets translated into an amino acid sequence. Remember, that's what proteins are. They're amino acids. So that amino acid chain, the primary protein, gets kind of packaged up in here. And then it goes to the Golgi body. So it's getting packaged. It goes to the Golgi body. The Golgi body kind of reworks it a little bit and then ultimately packages it back up to where it ultimately needs to go. In this case, it would probably be leaving the cell. So it's super complicated. There's all sorts of things going on. So all these parts of the cell play a role, right? So cells that make a bunch of stuff are going to have lots and lots of Golgi bodies. If the cell doesn't make as much stuff, it's not going to have as much. All right, so again, nucleus, endoplasmic reticulum, bumpy endoplasmic reticulum, smooth. Smooth makes lipids, bumpy makes proteins because of the ribosomes. Golgi body, and then all the way out. All right, now come the mitochondria, which are so cool. And when we learn about how we make energy, we're going to talk tons about the mitochondria. But for now, all we really need to know is that mitochondria is the place in the cell where we make energy. So oftentimes the mitochondria is referred to as the powerhouse of the cell or the energy factory of the cell, and it's making energy. And of course, we know that word energy actually means that it's making adenosine triphosphate, or ATP. ATP is made in the mitochondria. And the mitochondria are kind of crazy. I think they look like peanuts myself. Mitochondria are oval shaped. They've got a double membrane, which we'll talk more about later on. And each membrane has a phospholipid bilayer around it with proteins. And essentially what's going to end up happening is inside that mitochondria, we're going to make a bunch of ATP. We're going to make it out of sugar. We're going to make it out of fats. We're going to need a lot of oxygen to do it. And that whole process, of course, is called cellular respiration. So that requires mitochondria, intact mitochondria. So they're really important because they make energy. That's all you need to remember right now. So cells that do a lot of work, like muscle cells or brain cells or things like that, or kidney cells, they've got a ton of mitochondria. Cells that don't do that much work don't need to have quite as much in terms of mitochondria. All right, let's see what else. Do we have anything else? Oh, my favorite, the chloroplasts. So these are only in plant cells. No other cells have chloroplasts. Well, that's not exactly true. The plant-like protists have some ability to photosynthesize, but that's an exception to the rule. Plant cells have chloroplasts. Chloroplasts are where photosynthesis occurs, and photosynthesis is how the plant takes carbon dioxide from the atmosphere and water and the light energy and makes sugar. And oxygen is a byproduct. So unbelievably cool. That process is called photosynthesis, and you'll learn all about it later. So for now, all you have to remember is chloroplasts are sort of analogous to mitochondria, but you notice that the plant cell also has mitochondria. This is mitochondria next to it. So they also have their own mitochondria. Chloroplasts, and I should say this, chloroplasts have their own DNA, and mitochondria also has its own DNA, which is really weird, which we'll talk more about. And they play a role in photosynthesis. Okay, so plants and algae basically is where we see this happening. So anything that's able to make its own food is referred to as an autotroph. So plants are autotrophic. Chloroplasts structurally are a little bit similar to mitochondria. They have an inner membrane and an outer membrane. And again, we're going to learn all about those later on. All right, let's see if there's something else. Oh, the cytoskeleton. So the cytoskeleton, again, it's just kind of like a skeleton. It gives the plant structure. It's made of proteins. There's all sorts of different ones. It's really cool. It's stained in this picture, so you can see all the cytoskeleton. There's some other things that are sort of unique to certain types of cell. This is a paramecium, a single cell organism. It has cilia on the outside. It's like hair-like structures that helps this thing swim around. This is a bacterial cell with a flagella. It's a tail. There's only one kind of cell, animal cell, that has a flagella. Some of you might know what it is. It's a sperm cell, but bacteria have flagella, and those things whip around and help those critters to swim around. Okay, so that's it. That was a lot, though. So that takes us to the end of the cell and the organelles. Again, that was kind of just a quick introduction. Below these files, you're going to find a link to a couple of Bozeman videos, and then below that, you'll find an assignment. So make sure you have your study guide. When we come back to class in a week, actually a couple weeks, today's not quite your class day, but we're going to have a week off. When you come back, then you can bring your study guide, and anything that wasn't clear or you didn't get, we'll review in the beginning. And then we're going to do a couple in-class activities just to see how you're doing with this stuff. Okay, have a really great holiday, and I will see you soon.